it. Thanks everybody for being here. And I'll jump from here right into the introductions of our two panelists. So today we have Dr. Elaine Ingham, who discovered the soil food web nearly 4 decades ago and has been pioneering research ever since. And she is widely recognized as the world's foremost soil biologist, and she's passionate about empowering ordinary people to bring the soils in their community back to life. Dr. Elaine's soil food web approach has been used to successfully restore the ecological function of soils on 6 continents, and she has helped to make soil health knowledge available to people with no relevant experience and making it accessible to individuals who wish to retrain and to begin a meaningful and impactful career in the area that will help to secure the survival of humans and other species. So you can see the importance of what we're getting into here. And our other panelist, John Kempf, it's John's mission to have these regenerative models of agricultural management become the mainstream globally by 2040. In addition to being a grower, John is the founder of Advancing Eco-Agriculture, Crop Health Labs, is it pronounced Ozadia? And the Regenerative Agriculture Academy. He hosts the Regenerative Agriculture podcast, where he interviews top growers and scientists about the principles and practice of implementing regenerative agriculture at a large scale. So thank you both for being here. It's a great opportunity to speak to you again. And to start us off, I would like to pose the question, what is the potential that both of you see if we're able to reverse the trends of soil degradation through all forms of cultivation and stewardship? What would be the impact on the food system, the farming lifestyles, the climates, and our waterways if we're able to achieve this goal of repairing soils around the world? Perhaps starting with Elaine. You're still on mute, unfortunately.
There we go. Okay, so I didn't mute myself, so I didn't think I could unmute myself. Well, the answer to the question is kind of how far out do you want to go? Because what's the timeline? Because realistically, if we start adopting the kinds of things that John and I both talk about, we're going to live in Eden. It's back to the garden. You walk outside and you take the fruit off the trees. You have your garden. It's right there, and it's got all the plants have maximum nutrition that they possibly can hold, because every second of every day with good healthy food web in your soil functioning for that plant is going to get every single nutrient properly balanced for that plant, because you have the biology in the soil up and running the way it's supposed to. You balance the fungal to bacterial ratio, you've got the right amount of predators, and so that plant's getting all the nutrients it needs. It's protected above and below ground by all these microorganisms on those surfaces. The pests and diseases, the parasites don't even know the plant's there because it's so well protected. The roots go down as deep as they can, and structure has been formed in the soil. There aren't any compaction layers. The plant can get to that summer water stored by snowmelt or by rainfall in the springtime. Us as human beings don't really have to do anything more than plant the seed. What do you want to put in here this summer? What do you want to actually grow? And so the plants, the seeds, the starts go into the soil. And other than that, once you have that biology back in the soil and it's working full time and you're feeding it by having understory plants present in the system, you can go fishing for the rest of the summer, or whatever you care to do, hiking in the mountains. You do have to come back every once in a while and check and make sure that there's no little surprises starting to happen. But it's almost, from my point of view, that's a perfect lifestyle. You don't have to get involved in stressful, nasty human interactions where we're fighting over something that we shouldn't fight over in the first place. Abundance is easy to come by if you start understanding what you're doing with that biology, with the plants, with the animals. And yep, it's in systems that have to be understood completely so you can tell when something's wrong. You have to have that microscope to be able to tell you when something is starting to go wrong. By the time you've got dead and dying plants, you messed up. You didn't pay attention to what Mother Nature was trying to tell you. And so she's going to visit death and destruction upon your garden. So we need to learn all those signals. We need to learn how to read that book of Mother Nature. So I've probably gone on and on for way too long. So I'll stop here.
No, it's okay. That's a wonderful vision. John, can you tell us what you see as a vision for healthy soils around the world, especially in the agricultural context where you're used to working?
I'd like to expand the conversation beyond just healthy soils. I think Elaine mentioned this and described it very well, but rather than focusing exclusively on a conversation about a healthy soil food web, the ultimate expression of a healthy soil food web is a healthy life web. And Elaine, you described this very well. We need to also look at and consider the health of the plants that are growing on that landscape, the health of the livestock and the people that are consuming those plants. So to summarize it as succinctly as I can, I would say that nutritional integrity and microbiome integrity is at the foundation of health for the entire living ecosystem. And those are the things that we need to manage as farmers, as farm managers, as producers, whatever our role is in the larger ecosystem. And when you consider those two aspects and the future that can be brought about, I look at it from a very practical perspective. Is our agronomic management, our management of that microbiome and our management of the nutritional integrity, is that bringing about resistance to parasites in livestock and people? Is it bringing about disease resistance? Is it bringing about insect resistance? If the answers are yes, then those are, from a very practical foundational perspective, those are the outcomes that we seek and the outcomes that are possible. And if that is not happening, then we need to change our management systems.
Well said. And with that positive idea moving forward about what it is that we can achieve, there are some things, especially with how popular soil science and awareness around building biology in the soil has been in the last handful of years. There's also been a lot of misconceptions or misunderstandings about how to achieve this. John, can you speak to some of those that you've seen perhaps in popular culture and social media that are misleading or not really helping to advance a clear understanding about how to build healthy and biologically rich soils?
That's a very big, broad, open-ended question, Oliver, because there are so many misconceptions that it's hard to know where to begin. Perhaps I'll point out one that I've spoken to frequently, which is the idea that it takes healthy soil to grow healthy plants. And that isn't necessarily incorrect, but it most certainly is incomplete, because we need to ask the follow-up question of what is it that creates healthy soils? And what creates healthy soils is the contribution of plants. It is plants that contribute carbon. It's plants that contribute root exudates. And thus, it is plants that provide the food source for biology to thrive. So I've made the comment in the past that without the contribution of plants, soil is little more than decomposed rock particles, because there is nothing there for the biology to feed on. And so when we ask the question, if the idea is that it takes healthy soil to grow healthy plants, the next step, the immediate, obvious next question should be, what does it take to grow really healthy soils or to produce really healthy soils? And the answer to that question, in my opinion, is it takes really healthy plants and large populations of photosynthesizing plants to feed that soil biology. So I think that's one piece we need to consider. And then something else we need to consider is that, as Elaine has so eloquently described in her work, microbial populations have an incredible capacity to release nutrients from the existing soil mineral matrix and make them available to crops. And that is where plants and crops should be getting their nutrition. And we also need to consider the geological context. If we are working with soils that are based on a foundational bedrock that is completely absent some critical nutrients, we need to consider where those plants are going to source those nutrients from. So those are a few that come to mind. And I'll just add one last thought, which is that what we observe on large scale production agriculture is that most nutritional imbalances are not a result of what is absent in the soil. They are a result of the excessive fertilizers that farmers apply that create deficiencies of other elements.
Fantastic. I see some nodding in agreement and some questioning looks from Elaine's side. Can you give a rebuttal of what you just heard and maybe add some of the myths and misconceptions that you see out there?
Yeah, when we get into specifics, there's a lot of misconceptions, but I don't think we're really going to be talking at that scale today. Where making compost and the things that people mess up on. But when we're talking about healthy soil and healthy plants, it's a little bit kind of just dealing with grammar, maybe a little bit. But I want to take us back to the whole process of succession. When an ecosystem has been destroyed because of fire, extreme fire, or extreme flooding, the whole mountainside came down on you, and now there's just highly disturbed dirt. Most of the biology is gone. The first thing that comes back into that material, and it's really, I'm going to call it dirt because that's what it is. You don't have the food web in it, because of the disturbance factor. And yet the first thing that comes back into that system is not a plant. It's going to be microorganisms that are photosynthetic. And so they provide that first step into harnessing carbon, the energy of the sunlight, binding those two carbon atoms together, then the third atom, and then the fourth atom of carbon, fifth, and starting to build sugars. So, we start off right at the soil level of, we do need to understand how you get through that highly disturbed dirt stage of life. And it's going to be the microorganisms that are doing that work. Of course, as soon as you've got bacteria growing, now you're having some decomposition occurring, and along come the predators. You're going to be seeing the protozoa coming into the system pretty rapidly to eat those bacteria and start setting up nutrient cycling. It's not until you have enough of that nutrient cycling process occurring that rooted plants can come into that system. So it gets to be kind of a chicken and an egg question. What comes first, plants or the microorganisms that they feed? And then the microorganisms feed the plant back the nutrients they require. And what is it that really causes that successional process to shift? How do you get from just algae and lichens and moss growing on the surface of your dirt? What is it that allows those first weeds to start growing in your system? So you've got to accumulate enough bacteria, enough protozoa, enough bacterial feeding nematodes to start that process of nutrient cycling, or plants aren't possible. So now you have very weedy systems. That's not what we want for agriculture. Well, have to start getting the fungi back into the system so that you can have the NH4 in the soil. The ammonia must be present at certain levels, depending on what weeds you want to grow or what crop plants you want to grow. There's been a series of papers out of a number of universities in Japan where I went and talked with them many years ago at this point, but I think I kind of started them down this pathway of saying, okay, what's the balance of nitrate and ammonium? What effect does that have on plant production? And when nitrate is very high, that means you're going to be growing weeds, and just weeds. And if you have your crop plants in the system, ouch, it's going to be unpleasant until you can finally get ammonium starting to come into that system. And it's when that ammonium and that nitrate kind of shift in space. Now you can have highly productive shrubs and deciduous forests and eventually conifers. So that whole successional process, you've got to get fungi started. Well, what starts the fungi growing in these systems? They've got to have food. And so what provides that is indeed the plants, just as John said. If you understand this whole successional system in its entirety, what really starts everything is the biology in the soil. And then the plants can come in. And because those plants make cellulose, now we've got some fungal foods. Now the fungi can start growing. Now your plants can start making lignin because you've got the fungi there. And now the lignin is food to feed better, different kinds of species better, because from my point of view, because I like forests more than I like grasslands. What's more beautiful? Okay, I'm a child of the environment I grew up in, Minnesota, all those lakes and forests. Yeah, the most peaceful place I can imagine is sitting in the middle of a Minnesota mixed pine and deciduous forest and listening to the birds and the frogs and everything. That's my peace. That's where I go to meditate. So so nutrient cycling, all of that depends on a synergy. It's an interaction with both plants and microorganisms. So maybe we don't need to have that competition. We don't need to invoke that one's better than the other, or we're not going to have Eden if we don't put it all back together.
Wow. Thank you for not only explaining that, but also bringing me back to my childhood, because I grew up in Minnesota as well, with that idea in the background and the 15,000 lakes around that. That takes me back too. Now, John, in order to get a better understanding of your soil, you have to do some kind of analysis and some tests. And I know you work with growers all around the United States, and I'm sure have consulted in many other places too, about the best way to assess the state of health or degradation that your soil is in. And a lot of people are asking whether it's really worth getting professional lab tests, or if there's a lot of analysis that you can do on the site without having to go to higher tech methods to learn a lot about what's happening underneath the ground. What advice would you give on that front?
It depends on how dedicated you are and the outcomes that you desire and how rapidly you desire them, I suppose. So I actually gave an hour-long in-depth webinar on how our perspective on this has evolved over the last 15 years. That posted on YouTube just this last week, I think. I'm not even sure what the clip is called. But I believe that ultimately plants are the final report card. If we talk about disease and insect resistance, plants are the ultimate indicator of what is actually truly going on in the soil, and they are much more accurate than a laboratory analysis. But, and so in our work, in our professional work, we have worked with sap analysis very closely to evaluate how the nutrients, how the plants are absorbing nutrients from the soil profile. And that, of course, gives us indications into the soil's microbial activity, organic matter levels, and so forth. And so our kind of where we are at this moment, after collecting lots of data for the last decade and a half, is that we use sap analysis during the growing season to monitor the crop's nutrient absorption and performance. And we use, we still use the typical CEC soil analysis just because they are what many farmers are familiar with, but we have a growing distrust for them because we find that those nutrient analysis do not correlate with actual measured plant nutrient absorption, with the exception of a very few nutrients. There's no correlation. And so our work today, we usually pull, in terms of a nutrient assay, we pull a single geological analysis, like a mining analysis, mining extraction for each given soil type. That can tell us the total nutrient profile of what is in that soil. And you only need to do that one time. And that gives you an indication of the nutrient reserves that biology can access and can tap into and can make available to crops. And it's important to know this because some soil types, based on the geological bedrock foundational parent material, may not have certain nutrients that crops need, and get the majority may be there in abundance. So this is very valuable to actually see what the foundational parent material is that you have to work with. And then we do still continue to pull CEC analysis, largely just from a psychological grower comfort perspective, more than from an agronomic use perspective almost. We use them primarily for calcium, magnesium balance, and perhaps a few other things, but We're using them very minimally anymore. And then we're also using, just started in the last 2 years, widely using the Haney analysis, because that seems to correlate much more accurately with actual measured plant nutrient absorption. And it seems to connect with the biological capacity to deliver nutrients. And within the last year and going forward in this current growing season, we're anxious to do a lot more work with measuring biological profiles, because that has been a perceived weakness that we have observed in our own systems, that we haven't yet been comfortable with many of the microbial bioassays for repeatability and reliability. Obviously, your great work, Elaine, with microscope and microscopic analysis is foundational, but it's also user dependent, and many farmers don't have the bandwidth or the desire to engage with that. So that's something that we are actively evaluating and working on including more, and that's kind of where we are at the moment.
And Dr. Elaine, can you talk about some of the most important things that people should be trying to evaluate in their soils, and the best way, or perhaps even most economical way for growers to do that?
Yeah, we like to avoid the chemistry tests because they don't, just as John was saying, they don't really tell you about the potential pool of nutrients. They'll tell you what and typically the chemistry tests that you get from a chemistry lab is only going to be telling you about the soluble inorganic pool of nutrients. Maybe if you do a little work on calcium magnesium ratios, you're going to get a little bit about cation exchange capacity, but it doesn't tell you what the biology is going to be able to convert from plant not available into plant available. Knowing the organisms and the biomass of the bacteria and fungi are going to tell you a lot about that nutrient cycling process, the potential. If you then also add the protozoa, the nematodes, the microarthropods into that biological assessment of your soil, you'll get to understand the rate at which those nutrients are going to be made available. And of course, it's all of the organisms together that allow you to understand the balance, that you're getting the balance of all of the nutrients, not just nitrogen or phosphorus or sulfur or magnesium, but everything. And your plant requires all of those nutrients. Quite often, a plant is actually going to be restricted from being able to grow because of some minor, or we call them micronutrients. Still means that you've got to have that micronutrient present in adequate amounts. And most of the time when I look at parent material and try to understand what are the nutrients that are going to be made available or can be made available, we're going to be able to assess that because it's the biology that is causing that shift into plant available. And in fact, I don't know of any parent material that lacks any of the nutrients that your plant requires. There's some strange marine sands that have been sitting at the bottom of the ocean for the last, 10 billion years. Well, maybe not that long, but for a very long time. And by the time they're scraped up against the shoreline and they form dunes, that's not soil. That's dirt, that's sand. There's serious lacks in some of those things. They've got to get mixed into the parent material that the bacteria and fungi are breaking down on a daily basis. There's a huge amount of nutrient being released from rocks and pebbles and sand, silt, and clay and parent material, boulders, all of that. So nutrients aren't lacking in your soil. You may be lacking the microorganisms to convert them into an plant available form. And so that's what you need to know. So we encourage people to use a microscope, and especially for people who are not professionals. The idea is that we want you to take 1 teaspoon of your soil and mix in 4 teaspoons of water, shake it for 30 seconds. A drop goes on the microscope slide. You look at it. You don't have to quantify everything. So you want to get a good idea of the number of bacteria in that sample. What's the diversity of bacteria? Are you seeing lots of different sizes and shapes? Then you've got good diversity. Take a look at 10, 20, 30 of the fields of view on that microscope slide that you've put together, and how variable are those bacterial numbers? Well, be looking for fungi too. So we teach people how to measure what is identify what are fungal hyphae, what are the good guy fungal hyphae, what are the bad guy fungal hyphae. And they're fairly distinguishable. Things that fall into that omycete category, which taxonomists are arguing right now whether those are actually fungi at all. But I don't really care about that, because I know it's the omycetes that cause a great deal of the diseases. And that's what we need to be able to identify. If the only thing you're seeing in your soil is omycete, you should be worried right now. And you should do the things necessary to bring back in the biology that will take out those bad guys. Well, what if you've got the not desirable protozoa? You want to have just the good guy protozoa. How do you get the protozoa back into your soil? You've got to be making compost. There's your inoculum of all the organisms that you need. So long before the plant starts showing that there's something wrong down there in the soil and their roots have been massively attacked enough to start showing you above ground that there are problems, we want to be looking at the soil and fixing those problems before any problems ever start to occur. So that's chemical assessment of the soil. It's not telling you what you need to know soon enough. So if you can learn to just use the simple microscopy, have you seen any protozoa, any of the good guys in your soil as you're looking through your soil sample on the microscope? No, you've got a problem. If you're seeing a goodly number, okay, could be, you start feeding lots of bacteria and fungi, they're going to eat those bacteria and fungi, and now things may balance out. Just keep an eye on them as you go through the growing season, and you'll know in advance what's going wrong in the soil.
Well, given that we're kind of trying to suss out a lot of practical information, especially in these calls, because a lot of our audience are farmers and especially market gardeners, can you talk about some of the best ways to add that microbiome back into the soil if it's lacking, or how to build healthy soil, especially for that intensive annual vegetable production that a lot of our farmers are working with?
Yep. So in the fall, so when you start out next fall, please get a microscope, get a little bit of knowledge about what these different organisms look like so you can start to identify them. And that's what our foundation classes do for people. The fourth foundation course is all about teaching you how to use the microscope and how to identify these organisms. We don't have to go down to genus species, not by any means. We just want to put them into the basic feeding groups. So bacterial feeding, fungal feeding, bacterial decomposition, fungal decomposition. So we have a good idea of what you have in the soil. And if you see that you're lacking any of those organisms, then you're going to want to think about making compost. And wintertime is a great time to make compost because you don't have a whole lot of other things to do typically. If you're in tropical areas, it's during the dry time of the year. That's winter from a tropical point of view, is when you don't have enough water for anything to grow. What else do you have to do? Make compost. And so you want to collect your starting materials. We have certain recipes for the compost, depending on whether you're in arid or not arid parts of the world. So typically they're going to be around 10% high nitrogen. That's party food. That's gonna, in a thermal pile, that's gonna be what gets those bacteria and fungi growing so rapidly that they increase the temperature. When you think about one bacterium becoming 2 bacteria, there's a significant increase in temperature because of the heat of reproduction. So if you're getting 1,000 bacteria all reproducing in the same 20 minutes, how about 1,000,000? How about 10 million? You're definitely going to be increasing the temperature in that pile. And we want that temperature to get above 131 degrees Fahrenheit for a full 3 days, because you will kill all the weed seed. You'll kill all the seed in that plant material that you put into the pile. We want to be able to kill all of the human pathogens, all the animal pathogens. And because their cell wall structure is so different from the more environmentally survivable bacteria and fungi, the disease-causing bacteria and fungi are highly susceptible to those temperatures for fairly short periods of time. If you can get above 150 for 48 hours, 160, 65 for 24 hours, you're going to kill those pathogens and pests and parasites. They're dead. One of the few times I actually enjoy something dying is when we're dealing with the bad guys. What's growing? What's causing the temperature in that pile? And it drives me crazy when somebody says, now that your pile has reached 175 degrees, your pile is sterile. But how does the temperature remain high at 170, 175? How can it be maintained if you've got a sterile pile? So it's not sterile. There are lots of organisms that tolerate and grow and reproduce at those temperatures. When those organisms start running out of the simple foods, they've used them all up. So that's why we're putting in 30% green and 60% woody. Now you're going to have really good food for both the bacteria and fungi. Bacteria like the green stuff, fungi like the woody stuff. If your woody material doesn't decompose, is it that there's something wrong with my recipe? Or is it because you don't have the organisms that decompose that woody material? So there's a message from Mother Nature. You should have had put an inoculum of as high a diversity of fungi as you possibly could at the beginning of that composting process. You might be putting in some diseases and problem organisms if you're just going around collecting starting materials, but fine, that's why we're composting. That's why we're using a thermal composting process, so that the bad guys will be killed, the good guys will flourish and multiply, and you'll have massive diversity. Try to get as many different kinds of woody materials, brown materials as possible. The more diversity is going to result, because you get lots of different starting materials. Don't just put in wood chips from pine, put in wood chips from aspen and poplar and apple trees and everything you can possibly think of. A little bit of everything, because that increases diversity, and that means your soil is going to be able to deal with problems all the year long. So compost, thermal composting, we go through that whole process with you in our foundation courses. Well, what if you're not really into thermal composting? Because you got to pay attention to a thermal compost pile, because it is getting to be high temperature, and you better be watching and taking its temperature every day during the 10 to 15 days that it needs to be at high temperatures. As soon as it starts cooling off, then fine, you don't have to pay much attention to that pile at all. But what if, you just don't, your lifestyle doesn't, that's just not going to fit into your lifestyle? Then you make worm compost instead. It's the same kinds of starting materials, except you're not going to put the high nitrogen in, or you don't need to put the high nitrogen in. So let the worms do the job of changing the sets of microorganisms that are present in that plant material. And earthworms come with their whole back end of the worm is just a massive growth chamber for some of the best species of bacteria, fungi, protozoa, and nematodes that you can collect in your surroundings. Don't get the earthworms from just one little spot in the forest. Go out and take one worm from here and another worm from over there and another worm and another worm and another worm. There's some really interesting work by David Johnson where he's been looking at the species of bacteria when you inoculate just one population of earthworm, you only have the same set of microorganisms in all your worm pins. And that means a limited diversity. So there has to be, now that we know this, we have to start saying, don't just get one batch of earthworm. You want to get, maybe 10 or 20. So go to everybody else's worm bin pile and take a little bit with you and get those organisms inoculated into the pile. So I don't know if I've answered all of your question, but I think I've probably run out of my fair share of the time here.
No, you're fine. Those are all great suggestions. John, same to you. What recommendations do you give, especially to intensive annual vegetable growers, on how they can provide the adequate nutrition in their soil to have a successful growing season?
Building on what you just described, Elaine, I think compost, vermicompost, or thermal compost, different, also Johnson Sioux bioreactors, compost is a very valuable and very necessary contribution in these very intensively managed production ecosystems for market gardens and so forth. In our experience, I would suggest 2 additional pieces that we should consider adding to that. One of them is purchased inoculants. And my experience with purchased inoculants comes as a result of, I guess, let me say it this way. We know that we have, my understanding is that we have very rapid exchange of genetic information between different groups of soil microbial communities. And we live in an environment today where, depending on where compost materials are coming from, we get exposure to lots of different pesticides and lots of hydrocarbons. And it's possible for us to get microbial inoculants that have the genetic information to bioremediate those hydrocarbons very quickly. And our experience has been that when we apply these types of biology to agricultural soils, we get surprisingly strong crop responses. So we're evaluating the effectiveness of these materials based on crop response, and the crop response has been really extraordinary. So I think that's something that we should really consider and keep our eyes open for. And then I would also add that while compost is a very valuable, or inoculants are very valuable and I believe necessary contributions, we also need to remember that there are many soil microbial populations that can only thrive and reproduce in the presence of a living root system. Mycorrhizal fungi are perhaps the classical example. There's many others as well. And so I would suggest that one other piece that we need to consider is we need to grow diverse crops and cover crops that can feed an abundant microbial population. And that is also foundational to really regenerating our soil's microbial community. So I was asked to do a presentation a couple of months ago for the Bionutrient Food Association, where I was asked to put together kind of these high-level principles of how do we develop these market garden soils to very high levels and very high plateaus of performance. And my recommendation was that fundamentally we need to consider the living microbial zone in the soil, whether that's a depth of 4 inches or 20 inches. We need to manage that as if though we were managing a petri dish in a laboratory, or you could say as if though you were managing a sheet composting pile. So that means that you cannot let it be exposed to the sun, because when you expose it to the sun, high levels of oxidizing radiation, that is going to shut down microbial populations. And you need to manage water, particularly excessive water. So we live in a very different world today from what and we need to acknowledge that we have vagaries of the climate that are much more severe than they were even a decade or 2 decades ago. So soil biology can recover quite nicely from drying out. And obviously it also recovers from being saturated. But our experience has been that in these biologically managed systems, having saturated soils for extended periods of time really does a number on the soil biological population and on crop performance. It's much more damaging than having soils dry out. And so I believe it is of critical importance for us that we need to develop our rainfall management in the landscape to the point where we have the capacity to handle 3 to 5 inches of rain in 24 hours. That seems to be happening multiple times per season in many parts of the world. So whether that means very shallow subsurface irrigation, whether that means contouring to move surface water off, whatever it is that means for the local landscape, that piece needs to be figured out, because keeping soil saturated for extended periods of time is a significant problem for that petri dish in the microbially active zone. So those are the thoughts that I would add.
Well, while we're on the topic of soil amendments, and seeing as it's a large part of the business that you run too, is the inoculants for kickstarting the function of the ecology. Can you tell us about some of those inoculants that you found most successful and most applicable on a broad sense, but again, especially towards the market gardening or vegetable production side, just because it's a large portion of our of our farmers at the moment?
Yeah, I didn't really come here to do an infomercial, and I guess I'm really not actually when I start to think about it, because we actually don't produce any microbial inoculants on our own. So what we use commercially is we use mycorrhizal fungi inoculants on just about every time a seed is planted, and particularly for annual crops. And if you have a perennial ecosystem or crops that have living roots present all the time, the intention and the goal should be to inoculate a population of mycorrhizal fungi and other fungi that does not need to be constantly re-inoculated. And so we've certainly been successful with that. We often inoculate only for the first year or two, and then expect that population to maintain itself. And then we also use microbial inoculants from Tineo Technology. And the reason we use that company's products is because 10 years ago, 15 years ago, when we first started, we did extensive research in the field, comparing different companies' products. And when we look at purchased inoculants, I'm sure Elaine could give us a long list of possible downsides, and they do have potential possible, I shouldn't say downsides, but potential lack of effectiveness, because their effectiveness is really context dependent. It depends on where you put them and how they're managed. And the question that should be asked of those people who manufacture microbial inoculants is not how much does it improve crop performance, how much does it improve yield, or whatever the case might be. Instead, the question is, what percentage of the time do you observe crop results or not observe crop results? That's a much better question to ask, because in fact, it's a better question to ask not just of microbial inoculants, but of biological practices in general, because the reality is that the results are really context dependent of what's happening in that local geology and in that local landscape.
Okay, and Elaine, perhaps expanding more on the compost side of things, since you're very well known for that specialty, there's been a number of questions about different forms of compost, especially different anaerobic methods and aerobic methods like bokashi and others. Could you say a few words on the applications and the different efficacies of those different types?
Well, it's I think we probably have a little controversy going with Korean natural farming with the use of the anaerobic compost teas. And it's not that anaerobic compost teas don't have a role and function. You just need to know what that role and function is. You go and purposely make something anaerobic. The aerobic organisms that were on those bits and pieces of soil, and organic matter in that compost tea, they're going to either, their enzymes only function when the oxygen is at a certain level or above. As that oxygen starts to get restrictive for the aerobic organisms, they're either going to die if the loss of oxygen is very rapid. Many of those bacteria or fungi or protozoa or nematodes are going to just out and out die because it's too fast to change for them. If there's enough time, they will produce spores. And so they'll make it out the other end. Whenever that material gets aerated again, those organisms will germinate and start to grow. So it's not like complete and total wipeout. You just have to control the system so that those aerobic organisms have enough time to produce the spores, the dormant stages. So now you've dropped down into anaerobic below 6 pound ppm, and your anaerobic organisms are starting to wake up because now their enzymes are working. They're functioning in the atmosphere that lacks oxygen. And now they're capable of taking all of these, you know, proteins and amino acids and sugars and lipopolysaccharides, et cetera, and converting that into more bacteria. And they're releasing waste products that are typically organic acids. A lot of them are volatile organic acids. You're going to lose your nitrogen into the atmosphere because those bacteria have converted the NO3 or the NH4 into NH2. So you're losing, and it's not NH2, it's N2, sorry. Too many things going through my head all at the same time. So you're going to lose that nitrogen as a gas, as ammonia, NH3. With your sulfur in the aerated forms, SO4, SO3, SO2. So all of your sulfur compounds in your soils will be converted, or in that compost tea will be converted into H2S, hydrogen sulfide, which smells like rotten eggs. So you can detect when this is happening. You can detect the smell of ammonia, and you're losing nutrients. So now what good is it that for you to be putting that compost tea on your soil, because you've lost the very nutrients that you wanted to have get into your soil. You're producing organic acids. You're producing acetic acid vinegar is a very common one. Lactic acid is another very common one. A whole slew of them. And you can watch your pH drop in your compost tea from, you might have started out at 7, 7.5, it starts to drop down to 6.5, 5, 5.5, 4. I have seen compost teas that have ended up with a pH around 3. So imagine what you're doing when you're taking a liquid that is that low in pH, that acidic, and now you're spraying it on your plants. You can kill a lot of stuff with that. Well, it's really a pesticide now, isn't it? It's not a positive nutrient fertilizer. It's now something that is so acidic that you're going to cause some trouble, perhaps. And we've seen that sort of problem with the burning marks around the edges of the leaves when you let that get too anaerobic. So what if you don't let it get that anaerobic? Well, it's a really good fungicide. So you can put those organic acids out, and you can really knock back something like a verticillium or a pythium or a phytophthora infection on the surfaces of your plants or down in the root system. So you're going to wipe out a lot of those organisms. So you can have a beneficial effect from that. But by applying all of those organic acids to your soil, you've killed a lot of the beneficials as well. So how much damage has been done to the disease-causing organisms? Yay! How much damage has been done to the beneficial organisms they should be there to help your system recover? Well, you killed them. So if you don't do anything, what's going to come back into the soil on its own? In about 2 to maybe 3 weeks, what you find is that the disease-causing organisms have recovered more rapidly because that's their life history strategy. If just one cell survives, it's going to become 6 million cells by tomorrow, and the day after it's going to be 1 billion cells, and the day after, yep, now you've got the disease coming right back at you, or another disease coming along behind the first one that you hopefully got rid of. So if you do use those anaerobic teas, let me recommend to you that the way to allow you to use the anaerobic teas is to immediately come back with a good compost, a fully aerobic compost tea, where you have all the active, growing, living, healthy, happy aerobes in that system, who can rebuild the structure in the soil, who can deal with those organic acids and convert them back into bacterial and fungal biomass. So it's not like it, you would never think to do this. No, that's not what I said. Know what you're doing. Know what's the consequence of altering the biology like this. And, where you have those kind of specific questions, give me a call, or, info@soilfoodweb.com to, get some of your questions answered. Yes, we are going to charge you for our time answering your questions. Sorry, we've got to stay alive too. I have to pay all these salaries. So understanding, and that's really what I want people to do, is have an understanding of the consequences of what they're doing. And is it anaerobic or aerobic? And that's like what John was talking about, where you have water sitting in your field for weeks on end. If the flooding is not infiltrated, well, why isn't the water going into your soil? Because you got a compaction layer, and there's no way that water can drain. Eventually it's going to either evaporate or it's going to go downhill. And when that water goes downhill, it's taking a lot of your topsoil with it. You don't really want that to happen. You want infiltration. What builds infiltration? What takes out the compaction and turns it back into good fluffy soil that you don't have to fluff? Well, it is the biology. So get the biology out there. Well, it's not doing the job yet. Okay, you don't have any understory plants. You've got to get out plants that are present and growing all year round. You really want something that's going to green up and start growing in the springtime before your crop goes in. Yep, you can have furrows, and you drop your seed into the furrows, maybe along with a little microbial inoculum of all of the species, not just one or two species of bacteria, one or two species of fungi. Let's get the whole diversity. So you're not going to have growth just for a few weeks in the summertime. You're going to have growth aided and abetted by these really beneficial organisms throughout the whole entire growing season. So let's get that biology out there. If you've got permanent understory plants, they're perennials. You only have to buy the seeds for them once in your entire lifetime. And then those understory plants will protect that soil surface from the impact of rain on the surface of the soil that is so destructive. In some instances, rainfall is the most compacting thing you can get. Forget your tractor, forget your animals. It's rainfall if you've got bare soil. So get some vegetation, mulch if nothing else, to protect that soil surface and allow better infiltration. Don't allow that compaction. And usually people say, well, the rain doesn't compact my soil. Yes, it does. It just doesn't do it at the surface. It may do it down at 3 inches or 6 inches or 10 inches, depending on all kinds of factors. So I could go on, but I'll break off now.
No, this is great. And both from your comments and John's, we've had just an influx of extra questions, and people are really engaged about these topics. I'm really thrilled about the reaction here. But since we focus so much on market gardening context for the most part up until now, let's talk briefly before we hand it over to listener questions about broader acreage applications. And there's a lot of talk between the most effective and the most, economical way of regenerating soils on broad acreage, whether it's cover crops, whether it's rotational grazing, whether it's a mix of many different methods. John, can you start by talking about some of the results that you've seen out in larger acreage applications about broad applications of regenerating soil at an effective and economic scale?
I'm very passionate about having regenerative agriculture become the status quo globally in the next 2 decades. And so I'm very deliberate about thinking about how do we get there from here? How do we achieve the point where we have 85% adoption globally by 2040? And I believe that if you want to achieve anything, you achieve what you incentivize. So it's very important for any types of recommendations that we make if we want to have rapid adoption, we need to show an economic response immediately, not deferred 3 years, 5 years down the road, but there needs to be an immediate economic response. And so this has been the foundation, one of the foundational thesis of our work at Advancing Eco-Agriculture. And we have observed when we constantly are evaluating different types of recommendations, and the two recommendations that produce the most immediate economic response that facilitate the transition to regenerative agriculture are, number one is inoculants applied at planting as a seed treatment. They are relatively inexpensive, usually costing a few dollars per acre. And while not always effective, they are effective consistently enough, often enough, that they deliver very strong ROI, and they help facilitate that transition to a more regenerative approach. So that's one. The second most significant economic crop response is from the use of properly designed, and that's a key phrase, properly designed foliar applications of nutrients that are specifically designed to increase a plant's photosynthetic performance. And this is a key aspect, a key element that is completely missing from all that I have seen from all of our carbon sequestration research. It is assumed that photosynthesis is a constant. It's not. Photosynthesis is not a constant. Photosynthesis does not happen at a constant rate. Photosynthesis is highly variable based on the concentrations of carbon dioxide in the atmosphere, based on sunlight, based on available water, and all these other things. And what we consider to be normal, what we consider to be common, is crops that are photosynthesizing somewhere in the neighborhood of about 15 to 20% of their inherent photosynthetic capacity. It's not reasonable to anticipate that we'll get to 100% capacity in an outdoor agriculture environment, because that would require an ideal environment with an ideal context. It would require everything to be optimal. That isn't likely to happen on a continual basis in most outdoor production agriculture, but it is reasonable to expect that we can get to 40% or 60% of photosynthetic capacity, which would mean that we get 2 to 3 times more sugar production in each 24-hour photo period than what is presently common. Now, when you think about the implications of that, what does that mean for carbon sequestration? And that is never considered in any of the research that is being done on the capacity of agriculture ecosystems for carbon sequestration. So anyway, getting back to the focus of your question, our observation has been that when foliar sprays are properly designed to increase that plant's photosynthetic capacity, plants capture a lot more carbon dioxide, they capture a lot more solar energy. Photosynthesis is really the only way we have of bringing new energy into the ecosystem. So when we spike photosynthesis, that doesn't just mean we get more plant yield and more growth that offers an economic incentive to growers. It also means that we transmit much larger quantities of root exudates into the soil microbiome to feed soil biology, and we build biology much faster than we do with a normal crop. So, and I'm using this word normal or common in the context. We should ask what really is normal. Most of us don't really have any idea what healthy plants actually look like anymore. We think that a healthy plant is a plant that might have susceptibility to diseases and insects. Well, if you have diseases and insects, you do not have a healthy plant. It's really that simple. You do not have a plant that is photosynthesizing at optimal efficiency. And so I think it is worth pointing out, we have been framing this conversation up to this point largely in terms of managing biology, which is extremely important. But increasing photosynthesis and increasing the quality and the health of an overall ecosystem, it has a foundation on 2 pieces: microbiome integrity and nutritional integrity. Those are certainly tied together. They're certainly synergistic, but because you have one does not necessarily mean you have the other. And in fact, in the case of, as Elaine pointed out earlier, you can have perfectly balanced chemistry in the soil according to a laboratory assay, but still have unhealthy plants. So we need to manage both of those systems very well. So anyway, we begin when we first begin working with growers who want to make a transition on a large scale, I'm talking a scale of thousands or tens of thousands of acres, who want to make a transition to regenerative agriculture practices, we begin by looking at the cultural management practices and the use of products that will offer an immediate economic return this year. And then we compound that. And we often don't begin with large compost applications and the intensive use of cover crops in the first year. That is usually happens in year 2 or year 3, depending, again, of course, on context and local availability of resources. So everything is context dependent. All of these different practices that fit into regenerative principles are useful and appropriate. Some of them are just a lot more useful and more appropriate at specific times in a specific context than others.
Marvelous. Some great considerations there. And Elaine, I want to give you a chance to kind of review what was said there and expand on it. But I also really want to give our listeners a chance to ask some of their own questions, because there's just been such an amazing response in the chat here as well. And quickly, before we transition over to that, I want to let everybody know that we've got a survey that we would really love people to fill out here so that we could continue to provide educational opportunities like this. And you can give us ideas of who you want to hear more from and topics that you would like us to cover. Ivo, if you could put that link in the chat so that everybody could get a chance to give us the feedback that we can continue to make this a resource for all of you growers out there. And with that said, I want to start the listener questions with one that I promised to get to a second ago by Alexander, who writes, and he's a market gardener as well. How do they estimate the risk of eutrophication when using deep mulch compost? Obviously, in his market garden context, he's in a water protection area and is a bit worried about regulation coming in and possibly shutting him down. Can someone speak to that?
I'm not really sure what. Yeah, I'm confused, puzzled by exactly what that is that you brought up.
If I understand it correctly, it's worrying about nutrients leaching into the waterway. He's near a river, and there are tight regulations on that in Germany. And though it's not synthetic nutrients, he's not using fertilizer or anything, the amount of nutrients that are in that deep mulch composting could potentially be a risk, is what I've understood from that question.
Well, we don't ever see that problem with compost, because if you have a good, healthy food web in there, the bacteria and the fungi are going to be retaining all of those nutrients. It is the bacteria and fungi directed by exudates from the plants that the plant tells the bacteria and fungi what enzyme it should make, because the plant is controlling the kind of food resource that's being made available to the bacteria and fungi. And so the bacteria and fungi make those enzymes, pull those nutrients into their bodies. None of that is being put out into the soil solution. It does not leach, it does not leave the system. When the protozoa and the nematodes and the microarthropods come into the root system, attracted by the fact that there's massive numbers of bacteria and fungi growing around those root systems, then because the concentration of nutrients inside the bacteria and fungi are so much greater than what the predators need, the predators will release those soluble inorganic and organic nutrients as, call it their poop, if you will, the poop loop. So there's the soluble nutrients, but you're right there next to the root system of the plant, and those soluble nutrients are going to be taken up immediately by that plant. What the plant doesn't take up is going to be pulled back into the bodies of the bacteria and fungi that didn't get eaten by the protozoa, nematodes, and microarthropods. So there's, the interaction there, trying to follow, where's the nitrogen going? Where's the sulfur going? Where's the phosphorus? It's just so complex. And like Suzanne Sumard, some of her, yes, we just finished the soil summit in, well, actually today's the last day, soil summit at Soil Food Web Incorporated. And in her talk, it was really quite interesting as they're mapping out all the connections and all the interactions below ground. It was marvelous to see. So we don't have nutrients, soluble nutrients are not being made available. If you've got the right balance in your microorganisms, you should never be losing nutrients through leaching. It doesn't happen. And time again, work by Gabe Brown and Ray Archuleta has shown that you get the proper biology back into the soil and you have no leaching losses. None of that soluble nutrients stays around long enough in that soil to be leached. Something's going to take it up. And that's why both John and I are so much in agreement. You don't ever want bare soil. You want roots, you want plants growing those root systems down. I just choose to get perennial plants and get all of the soil covered and keep it covered all year round, if at all possible. You know, what we usually first do is go out and get like 25 different species of the short, low growing plants that we think will work in your area and just broadcast them. And what comes up, comes up. You typically get pretty good coverage within a year or so. If you have bare spots, you go and put some hay or mulch out on those spots and mix a few seeds in so that you can get that complete coverage of your soil. And then, you know, long before your crop is going into the soil, your cover crop, your cover plants, they're not cover crops. I'm sorry, cover crops don't fit the bill because cover crops are typically annual. You have to pay for that seed every year. And now once the plant is started to grow and is getting really healthy, now you got to mow it down somehow. That just is, it's nonsensical to me. Pay once for perennial plants, get a good diversity so it doesn't matter what this summer is going to be like. You'll have cover plants that will survive. And so long into the fall period, way after you've harvested, you've got plants in the field keeping those microorganisms alive and happy and being ready for whenever you do put your crop in. I do like to, just one little thing I'd like to add is we work with very large scale growers. So one of our projects in Southeast Asia is 50,000 hectares. So we're making the proper biology to go out into that system. And we're doing a pretty good job at it.
Excellent.
I'll add one thought regarding eutrophication, just kind of a very simplistic perspective on what Elaine described. Fundamentally, it comes down to managing carbon and nitrogen ratios. So if you're using animal manure-based compost that does not have enough carbon, then you might suffer potential challenges from leaching. But if you have enough carbon in the system, that will not be a problem.
Does that apply to additions like biochar? I know there's been some questions around that too.
I have a contrarian opinion about biochar that is based on lots of field experience, where we have actually evaluated biochars or the response to biochar of different microbial populations. And we're measuring this specifically as a combination of microbial population response combined with crop response. And we get peak responses. We do see responses and improvements in soil fungal populations and so forth, but we get peak responses at about 100 pounds per acre and no additional responses at higher application rates beyond that point. And that's based on testing on many acres on many tests. So I'm not sold on the hype.
I have to agree with John that I think there was a large amount of hype involved. And you know, when it's come down to actually doing replicated scientific trials, like at Rodale, our soil at Rodale was already well structured, and so addition of the biochar didn't do anything. And we applied at like 3 or 4 different concentrations, and we had controls where we didn't add anything, and then increasing amounts of the biochar, and there was no benefit, because I think in large part what the biochar actually does is improves fluff. It allows oxygen moving into the soil in materials that perhaps were completely compacted. So there's a benefit there. But you got to remember biochar, it has been cooked at a very high temperature and it's sterile when the biochar comes out. Where's the bio in that char? You have to add that biology back in.
I'll add one more piece, which is that biochar is not nearly as effective at sequestering carbon as high photosynthetic efficiency plants. When you calculate the quantity of carbon being sequestered by plants that are in a healthy photosynthetic state, it exceeds by orders of magnitude what can be accomplished with biochar.
It's good to have that data. Great.
Yep.
Very nice. All right, we're going to move on to another question here for both Elaine and John from Lior, who asks, what do the particular plant families that show up voluntarily tell us about the soil? I assume this is during the recovery portion of the succession. For example, brassicas, legumes, etc. Perhaps, John, you want to start with that?
Well, there's no way to answer that question in detail in a short amount of time. I would just say it very simply. I'm sure Elaine can expand on this. I would say it very simply that they are indicators of the microbial populations and the nutritional profiles and the physical structure, not just one by itself, but all three in combination. And so they are expressing where that soil is in its successional state. And it is worth pointing out that soils can be managed and can be balanced so that our domesticated crop species are healthier on those soils than these pioneering plants that we call weeds. And when that happens, diseases and insects will attack the weeds first and leave the crops alone. It's a very common phenomenon. Lots of growers have observed it. And that is an expression of soil physical structure management, microbiome management, and nutritional management. It takes the combination of putting all those three pieces together in the soil profile to achieve those types of outcomes consistently over time.
So, from my response to that kind of question is, if what is growing in your soil is a whole bunch of weeds, it's not really soil yet. So you're missing something. And I would want to look at what you're missing so you could move things along into a later stage of succession, something that you're going to be able to get some info. You'll be able to put that biology in and get the soil growing more later successional sets. It's my phone going off and I can't reach it, so we're just gonna have to listen to it. So when you're looking at legumes, for example, it depends on exactly which legume you're working with, but say something like alfalfa, it requires a 1-to-1 ratio, fungal to bacterial ratio in most situations. And so if you've got the alfalfa, that's what's coming up, that's what's growing, that kind of tells you what your fungal to bacterial biomass ratio is. And especially for good, healthy alfalfa, you need to have at least 300 or 400 micrograms of fungal and bacterial biomass in that soil. You better have the protozoa, and you should have the nematodes, mycorrhizal fungi should be there. So if it's good, healthy production, you're balancing the biology to grow that plant. And that's typically what we then find in the soil. If those are not healthy alfalfa, then you're earlier in succession. You've got too much bacteria, not enough fungi typically. So again, look at your soil and figure out why, what it is you're lacking so that you can fix that problem.
Fantastic. All right, next question is from Alba, who writes, we wonder how to deal with the accumulation of phosphorus in soil when applying big quantities of compost as mulch around 8 centimeters of depth in a no-till market garden system. We use compost as mulch because it is the most efficient system in our human scale garden. I guess that's not exactly phrased as a question, but she's asking about the accumulation of phosphorus in that kind of soil enrichment system.
It suggests to me that they're putting in manure that's extremely high in phosphorus. The animals are being overfed with a phosphorus substitute of some kind. And the animals are just going to pass right through their digestive system. Unfortunately, it causes damage in their digestive system, typically, that high a level of phosphorus. They're going to get rid of it through the poop. And so the manure that people are using to make the compost are probably having troubles with things composting correctly because the phosphorus is so out of balance. Quite often, if you've got a phosphorus, too high level phosphorus, your potassium is going to be really high. Your cation exchange capacity is going to be just crazy. So you're going to want to use less of that particular manure because you've got to dilute the amount of phosphorus by the amount of organic material you're adding that manure into. So they might need to be looking for a different source of high nitrogen instead of using that manure, or go back to the people providing that manure and say, could you please stop feeding the animals this real phosphate or potassium, whatever nutrient is out of balance, so that you're not feeding it to the animals. It's not good for their digestive system. It's not good for the soil, which is the Earth's digestive system.
Yeah, I would add to that and echo your concluding statement, Elaine, that fundamentally when you add excessive levels of nutrients, they cause all kinds of other nutrient imbalance problems. And phosphorus is a significant problem in this regard. When levels get high enough that it can even shut down populations of mycorrhizal fungi and other phosphorus solubilizing bacteria. The bottom line solution is don't apply such large quantities of compost that contain high phosphorus levels, and instead use compost that doesn't contain such high phosphorus levels and lower levels of manure. So I would say that has the potential to cause significant problems. We see it causing significant problems, and I would suggest just avoid it and don't create the problem in the first place.
Excellent. All right, I've got one more question for you here. I believe it's actually for your daughter, John. No, I'm just kidding.
She's so cute.
She's lovely. Okay, this is from Francisco, who asks, in your best client's farm, how many years did it take to stop using micronutrient foliar applications based on sap analysis to the point that the soil microbiome is already supplying all the nutrients the plant needs to build complex lipids, proteins, and unlock the active immunitary system of the plants? What I mean is when they reach their top health on the plant pyramid.
Well, that's actually two different questions. So to the first question of how rapidly can, I'll rephrase the question a little bit to offer some context. When you have soils that have degraded microbial communities, they generally have high levels of oxidized manganese, but don't have enough reduced manganese in the form that plants can actually absorb. And it is the function of beneficial soil biology, particularly many of the anaerobes that are in the soil particles, easy does it, little lady, that are responsible for converting this manganese to the reduced form in a form that the plants can actually absorb. That transition process of regenerating that soil biology to the point where it can deliver 100% of a crop's manganese requirement can happen in as little as 3 to 6 months in optimal environments. So that's the fastest that we've observed it. In some very highly oxidized environments like desert soils in the American Southwest, that might be a period of 3 to 4 years before that happens. It's very context dependent. So that was the first question. The second question was how rapidly can crops reach the top of the plant health pyramid? Again, it's very different if we're talking about a spinach crop that goes from seed to harvest in 37 days versus a crop that might take years to production. But it can happen in a matter of as rapidly as a few months to as quickly as 2 to 3 years.
And probably the only thing I would add to that is if we're using our microscopes to look at what the biology is in the soil, and you're seeing that things are getting balanced properly, we can usually do that in the first 6 months. Well, it depends on the crop again. So what's the length of that crop's life cycle? But even when we're putting in orchard trees, we usually can get the microbial balance back to a place that the trees are thriving, the cover plants that we have out between rows are thriving, the mycorrhizal network is coming together, and we get the mycorrhizal fungi reestablished within a very short period of time. If you already have the understory, the permanent cover on the surface of the soil, you put your orchard tree into the soil, and within 1 month, the network has formed and the nutrients are flowing in from the rest of the system to help all of those trees start becoming healthy and happy. So, we can manage things so much better when we have a tool that really taps into this microbial food web that has to be put back. It has to be what the plant requires.
Fantastic. All right. We've also received a number of questions about the Johnson-Su bioreactor. And instead of going through each one individually, I'd rather just let both of you sort of speak a little bit on it and its efficacy and its applications, and we'll wrap up in about 5 minutes.
So with the Johnson-Su bioreactor, it's basically, they're going to be doing a thermal composting to start off with, which is why they have those pipes that go into the system, so that, and lots of air holes in them, so that oxygen is maintained throughout the center, throughout the whole entire pile, when the microorganisms are growing very, very rapidly and using up oxygen very rapidly. They didn't want to have to turn. Well, so I've kidded them both about being lazy, because typically, you're out there with a pitchfork and a shovel. Well, but their system is working. Now, at 24 hours, they pull the pipes out because the chimneys have been established, and we've always used chimneys in our production of compost. So, where I just take a broom handle and put it down to the bottom of the compost in a pile like that. Or if we're doing windrows, you just go down the windrow and you punch chimneys into the compost where you're concerned that it might go anaerobic. And there are many times when I've been worried about that. So just punch holes, the chimneys in, and that works just fine. So they finish the thermal composting, making certain that things don't go anaerobic by not by turning, but by punching holes in the pile and maintaining oxygen conditions that way. They then come back in and apply earthworms into the reactor. And now all of that compost is recomposted by the earthworms, which are also adding all those wonderful sets of microorganisms that live inside the digestive systems of the earthworms. So again, go out to several different sources so you get more than just one community of microorganisms coming into the system when you add your earthworms. It takes about a year to finish a bioreactor, a Johnson-Su bioreactor. And most of the time with my clients, they don't want to wait. They don't want to wait that long for their compost to be done. When we compost using thermal systems, we're typically done in 30 days. So it's kind of, how organized are you in your life? Can you think to start a full year in advance? And I'm usually just not that efficient, I guess. So I like the 30-day composting process. And we can end up with very similar sets of microorganisms and diversity in all three systems like that. So the Johnson-Su is kind of a modified thermal composting, static composting system. And if that fits your lifestyle, that's what you should use.
From what we have experienced and observed, we have the benefit of working with, at this point, thousands of growers, mostly here in North America, but from some around the world that we get feedback from as well. And when we look at the performance of compost tea that is used either in furrow at planting or that is used as a foliar spray, our observation has been that the Johnson Sioux extract, I'm specifically talking about extract, seems to consistently outperform other composts that it is compared against. And I don't really have a dog in that fight because we're not really doing much work with composting in either direction there. So that's what our kind of observational experience has been. And I would just add one other thought that needs to be considered. There is this conversation around the need to, we had an earlier conversation about bokashi versus fermented compost and different types of composting. And we need to remember that there are many, the healthy soils will have vibrant populations of both aerobic and anaerobic organisms at the same time. That when we look at soil on a microscale event, when we look at within soil aggregates, inside soil aggregates, we actually have an anaerobic environment. And that we need to think, again, thinking about the soil microbial zone as a laboratory petri dish, we need to consider that our soil microbial populations live in an environment that at its peak, and in a healthy environment, is fundamentally a subaquatic environment where we have gas exchange and airflow on the outside of soil aggregates and soil particles. And we have actually an aquatic zone, an anaerobic zone on the inside of soil aggregates. So it is, I think, perhaps too confining to have a conversation, too limiting to have a conversation that says aerobic soils are good, but anaerobic soils are bad, because it's a lot more nuanced conversation than that.
Fantastic. All right, that's all the time that we have today. I really appreciate both of you and your perspectives and your knowledge on this. There's so much more to talk about these topics, as we've seen in the chats as well. Before we go, could I ask both of you to put in any links of how people can get in touch with you, see more of your resources? And I know, John, you've got a community as well that farmers can join. I'm going to quickly hand it over to Ivo, who's going to give us a little outro and actionable steps for our listeners in order to continue this conversation and participate. So thank you all so much.
Yes, thank you so much, Elaine, John. That was incredibly informative. And I think what we have seen with the questions, with the discussion, just how many, I mean, we know so much already about soils, and then there's so much that we don't know yet, and that we're still figuring out, and that we're understanding when exactly, where exactly, what. So really, really looking forward to continuing that conversation, collecting that knowledge. And I posted 2 links in the chat before for those of you that want to stay up to date to fill out. We also send that via mail afterwards, together with the recording somebody asked before. And there is a session next week where we're going to be basically mapping out what do we know about the transition from, let's say, a resource intensive production system to a regenerative one. So if you're a consultant, if that is something you're working with, you're very welcome to join us. And otherwise, I'll send you all the links afterwards to stay up to date. Thank you again so much, Elaine, John. That was a huge pleasure to have you.
It was great to be here.
Thank
you for inviting me.
All right. Have a great evening, everybody.
Thanks so much.
Take care.
Take care, everyone.
Wherever you are.
Ciao.